discussion, and I may not have answers, at least one of you will have some answer or add to the discussion in the context of speciation, EVO, DEVO, adaptation, selection, or that's one part. Let's see how much we can progress on when you get bored. Then I can go back to the PowerPoint again in the context of evolution, how one can study something in some aspect of biology. As an experimental biologist, I am only talking about the methods rather than the any specific.
It's surprising that these halters haven't evolved multiple number of times, considering how important, I mean, whatever the benefits that they provided to at least these particular group of insects. I mean, it evolved only once at the base of the dipteran lineage, right? How is it that it's not evolved?
Yeah, that's true, because see, I understand what you're talking about. So leg to wing transmission, the question is why halter have not evolved more than once? Because typically you have heard about, what is the word for that, the same type of morphogenic changes are convergent evolution, right? So leg to wing transformation has happened in insects, happened in birds, and happened in bats, right? 3 times, totally independent lineages. So why halter maybe only once, right? But in insect itself, the modification has happened multiple times. So for example, wing to elytra in coleopterans or wing to halter in dipterans, some modification, whether it is the same type modification, different modification. So why, I don't know, the one possible explanation is halter is still very recent, right, and it has happened in one particular lineage, that is dipteran, either one of the latest one. So the second is the modification. What other modification you can have it is the wing to halter in the forewing, that will change the flight pattern so much, it is unlikely that will. There is a insect called Strepsiptera, there is one where there is halter in the wing in the T3. It is a reverse, in a way it has happened more than once in the insect. Strepsipterans are another class of insect, they are not coleopterans nor dipterans, they are in another intermediate order, and they are also holometabolous. The Strepsipteran, they are an endogenous parasite. So in a way, the flight is I think compared to dipterans or lepidopterans, they do not fly around much. The larvae, when the pupate comes out, and then immediately it sort of lays on some other caterpillar or something, and then it dies. So that could be one of the reasons that while there is a potential, because it's such kind of changes, molecular changes have happened, wing to halter in the T2, but there's only 2 possibilities, and maybe it has happened only 2 times. At least your answer is yes, it has happened more than once.
Speed of the wing,
it's not been studied. It's very difficult to catch them and study. So I don't think there are any study on Strepsipteran wing beat frequencies and such kind of physiology and life history traits. There aren't much studies on Strepsipteran. Do you know anything about Strepsipteran?
Small wasps, the tiny wasps, the big wasps, they're really tiny, and they're known to utilize recurrence and help them move, and they can move over kilometers apparently by some estimates. So do you think these things probably, I mean, like, I don't know whether
for selection point of view or survival point of view. So the question is to how much it has to be a body change requirement, and how much it could be in the given environment it can somehow adapt because of the other non-biological phenomena. So let's say there are two purposes for which these locomotor organs. One is for foraging, the second is to avoid the predators, right. Now, avoiding the predators is basically a number game, right. You do not have to consciously avoid the predator, just increase the number, right. Instead of 1 or 2 progeny per female, you can have 200, 300, 1000, 2000, and irrespective of how the predators is, few will survive and the species will continue, population will continue. The second is the foraging too in a given even a large space, 1 or 2 square kilometer in the context of insects, right. There are small patches of availability of food and non-availability of food regions. If what smaller insects, right, if they have a body size is so small, they actually can get carried away with the wind rather than they have to have a directed motion with the help of all the energetics, and that can also sort of land them into the places where there is food. It's like wind pollination in plants, right. There is no direction. It's just that large number of pollens get sort of dispersed and then cross hybridization happens. Probability. So, in that context, the possibility that the body size and the halter evolution could be two independent events and may have converged in dipterans. There are some flies which are large. You must have seen a large house fly, and sometimes you wonder, is it really a fly or something else, right. Dragon, sorry sir, not a dragon, what do you call a monster, right. But anyway, so compared to other insects, dipterans, both small body size and the halter, one possibility is because it lost the wing, it could afford to have a smaller body size and move around, and or it, I don't know how to interpret the two.
Maybe due to the predation pressure it have, like if in the case of papilio butterflies, they have very low wing speed because they don't, most of the butterflies are toxic, so they don't have any fear of predation in that. But in the case of dipterans,
no, they have. There is also a counter evolution on the other side that you can have a detoxifying effect.
But
right?
What I'm saying that dipterans are more related, like more are pest or like more related.
No,
to
there are other papilio also,
yeah.
right? See, but the butterfly eye spot, for example, right? It's also to avoid the predators, but a completely nothing to do with toxicity. It is to sort of mimic, let's say, eye of a bird or something, so that certain kinds of predators may not come closer. It may actually predict. Sometimes the color is such that it may consider as a flower part of a petal rather than a butterfly. So there are many other type of mimicry there, camouflaging.
Intermediate where it's like not a halter nor a wing, like partial loss of function of Hox gene. Is it possible to create in lab?
We can. So, is interestingly, we've been talking about this in lab for quite some time, that halter is not a mini wing. So, in a way there is a clear change in the specification, it's not just about the growth control, right. But you can make slightly larger halter at very different stages. One of our undergraduate students in the 5th year project, not 5th year project, as part of his 2-3 semester projects, he tried this. Idea is to take different dipteran species, different Drosophila species, not really dipteran species. They have a different body size and the halter size, and proportion is slightly different. So, he actually did a lot of morphometric work to understand the differences, and then trying to put them through a flight assay and see whether there is any correlations. The statistics is not conclusive, we have to do many more work, but we still need to pursue that. And experimentally, what I was thinking, which has not worked, is simply because we don't have a good way of testing it, is now we think we can do is to create very different size and shape of the halter using genetic techniques in the Drosophila melanogaster and put them into a flight assay. Similarly, what this guy does. Sanjay, sorry.
So how much part of the Hox gene is really necessary or conserved part?
No, advantage is that's what. So we are only manipulating the organ. associated what our other physiology we are talking about, we are not touching it. Its its bioenergetics is not touched, its muscle patterns are not touched. So hopefully, if there is a direct correlation to the flight behavior and the halter size shape, right, then one possibility is there is a selection in one particular size and shape of the halter to have a balanced flight. It's not like what survived is what.
And how much part of the Hawkshin is like really functional, which is required? Is it the entire Hawkshin or just the small conserved part? Like suppose if we like
Okay,
create
so
some loss
this is
of
something
function.
what my boy here is trying to do. So idea is that this whole Hox protein what we tested, and only if you take the DNA binding domain, it will not work. That's for sure. What he's trying to do is to delete some specific elements and some making some mutations and see whether the UBX function is determined by the entire protein or some specific domains, or some are dispensable. That's what he's trying to look at. Right? That's what you're doing, right? Yeah.
So you showed that data like when advantage of two wings, having two wings, then four wings. So the flipping speed gets increased when the insect has two wings.
No, I didn't say get increased. I'm not giving any causal relationship. I said there is an association.
Yeah.
That the difference which fly faster or have a higher wing beat frequency compared to the 2-4 wing one.
Yes.
There's
So
no cause and
I am
effect
asking has anybody done such experiment to knock down the gene that is producing hind wing for a four wing things, and that is becoming halter. Then checking like that, then the mutated organism has only two wings. Now checking for that flipping speed, is it getting increased or not, like for a counter support of this?
No, there is no such experiment. I don't think so. First of all, only now we can manipulate other insects because of CRISPR-Cas9 technology. Until now, making transgenic and was very difficult. Some of the RNA approach was affecting the entire body so much. It was very difficult to make specific alteration to only certain specific organs. Now hopefully we can do such kind of experiment. You can take a butterfly or you can honeybee and make an halter in the T3 or just loss of wing, not even halter, and see how it affects.
Yeah, and if such thing happens, like if the wing flipping speed increase, then I will have the question like if it has so much of advantage, then why it is not so common?
Let's see.
Yeah.
So this is not pertaining to the current ongoing discussion, but you had mentioned the roles of Hox genes in limb development in reptiles, I think. So you said that the posterior Hox genes have more effect than the anterior ones, and you said that say the flank ones
Not
in the
more effect. Posterior ones are dominant over.
dominant, yeah. So you said the flank ones dominated over the anterior ones in these pythons and everything. That's why they don't have those forelimbs.
Yeah.
So can you extend the same argument to the say the tail, whatever is in the tail segment? Could you say that an overexpression of those Hox genes in the flank segment could lead to the development of an extra pair of limbs? Have somebody tried to do that?
So it has been tried in chick. So you can make 4-legged chick rather than 2 wing and 2 legs. But only obviously they won't survive because of so many other evolution, so many other things have happened. They won't come out of the eggshell. But you can actually see an embryo, the features of leg rather than wing when you do this experiment. It has been done. No? Any more? I think they look tired already. Yeah.
At the larger picture, like clusterstome and all, if you look at it, what really differentiates between two species? Is it forming a new north-south interaction, or the single gene cannot be the
Oh my God.
answer to it.
So this is
overall,
what you know, spending lakhs and lakhs or crores of rupees what you have been doing for the last 5 years. Okay, so what you are asking is the question about what is the consequence of UBX present and absent between T2 versus T3, and what is the what could be the differences between two species where UBX is expressed in T3 in both species, but one makes halter and other does not make halter, right. It must be something to do with the transcriptome. That is what we already hypothesized by exclusion rather than by actively determining it, right. If you remember the last slide I showed you, we think is supposed to be red. I do not know what you see here color. These are all black and these two are red. We said amongst the 5 possible ways in which Hox proteins can contribute to evolution of morphology in the context of wing versus halter, or when you compare T3 wing versus T3 halter in different insects, these 3 are excluded. Because these 3 are excluded, we think it could be happening at this level, and this you can study by ChIP-seq and by transcriptome. That is true. So, what we are trying to do is to identify genes by complete RNA-seq which are expressed in T2 versus T3 in all these insects, and also identify by ChIP-seq what are the targets of UBX which are directly bound on the chromatin in T3 of all these insects, and then do this network analysis. Yeah, it is a huge amount of analysis. We have done some work. I think it will be boring for everybody if you get into. We actually have done so many things, you know, in terms of what is a upregulator, what is downregulator, what kind of regulatory sequences by comparing 2 or 3 genes. For example, we have done for Apis, Bombyx and for Drosophila. Tribolium we are doing now. We have even done taken some of those regulatory sequences, made a transgenic reporter construct in Drosophila and even tested them in organism also. So, it is still an ongoing work. So, this is sort of So, what we are saying is there are some genes which are common targets in all the species, but for some reason the expression pattern different is because of some enhancer element which are different. It is not that UBX binding is affected, other transcription factor which interacts with UBX are different in different species, leading to change in the UBX function. Yeah. Yeah.
Sir, so most of the genes are negatively regulated, right? So for example, if there is no UBX, we will get a phenotype.
No. If you add UBX also you get a phenotype.
Yeah, but I am telling in a particular segment, we have an opposite effect, right?
I didn't understand. Say it again.
For example, if we have an ultrabithorax mutant, then it will lead to a wing. Yeah, in that particular segment.
Okay.
The same UBX is present where we need a wing. So if we have both mutant, it will not have any wing. So it's genetically programmed to make everything, and then it's reducing one by one. Right?
Oh yeah, yeah. Okay, yeah. I mean, you are a good development biologist. Yes. So, so what he is trying to say, which is true for almost all aspects. In a way, I think I thought I mentioned this in a passing remark when I was talking about the evolution of Hox itself. Let us say use this picture, right. What I mentioned was the earlier organisms, they had lesser features diverges along the body axis. When I say lesser feature divergence within an organism is like a earthworm, for example, you hardly see any difference from one end of the body to the other end. For example, if you take us or an insect, every part of the body looks different from the other part, right. So, there is a morphological diversity within an organism in insects or in human or in other vertebrates, whereas early organisms do not have that diversity. So, the morphological diversity within an organism is correlated to duplication and divergence of the Hox protein. In that context, I also mentioned that the posterior one are more dominant over the anterior one is simply because as and when new Hox is added, it basically modifies the existing developmental platform into something else. So, in that context, obviously you should have some developmental program. So, in a way it is true, although I did not get into the detail, the wing pattern, wing developmental program is what is known as ground state as far as the dorsal limb of T2 and T3 is concerned. UBX is modifying on it, whether to make an elytra or to make a halter, it modifies on it. Sorry, retain a wing in T3 in case of coleopterans. Yeah. What you are saying is, yeah. Anything else? If you have no questions, I thought I will talk about, just to entertain you, show some movies and leave you. So that, because I am supposed to speak until 6 o'clock, otherwise I can't take the honorarium. And so, shall I show you some movies then? Are there any questions? Okay. Okay. So, I do not have to tell you about all these things. Just about one aspect of evolution which I sort of forgot about before I start showing the movie, right. So, this is a standard statement you all know, otherwise you would not have been here. And you also know about this, right. These are all the basic features. You thought there may be certain what is known as invariant features, there are some variant features. Invariant features are the ground platform, you cannot tweak it too much. If you tweak too much, the cell dies and obviously there can't be any evolution because there is no survival, right, because it is so lethal. Any small changes to DNA, whole genetic information paradigm to transcription, translation, how the whole cycle, right. Now, there are some highly conserved functions, very interesting way, which is, we can discuss at length later if you are interested. If you look at this one, this is an insect, this is a compound eye, and this has some 700 ommatidia, and each ommatidia is made up of 8 photoreceptor neurons and a few other things, ommatidium, right? And this is the mouse eye and this is the human eye, and there are no retina like structure or lens like structure here. You have those kind of things here. Here the photoreceptor neurons directly send there is nothing like an image formation. The light falls and it sort of goes to the brain directly, and in the visual cortex of the insect sort of, and not actual the kind of image that we see will not be formed. What it look at is certain shape, size and the movement, and this of course forms a nice image, and that also forms a nice image, right. And a gene called PAC6, which is written here, a protein, right, in Drosophila is well known that it is required for formation of the eye. What it means is, as I told you in the hierarchy of gene expression, at somewhere in the embryo a few cells express a specific protein, which is this PAC6, and if they express PAC6, that means they are going to form the eye, right. And that is how the eye get positioned exactly in the same place, because of the whole regulatory network of very different genes and proteins will ensure that PAX is expressed here and nowhere else. Both has to happen. Otherwise, you will not be able to have this kind of a body plan, right? Not only that eyes should be formed here and it should not be formed anywhere else, right? Otherwise, you will get confused. Where are you seeing, what you are seeing, where you are going, and kind of stuff, right? So that means the hierarchy of gene regulatory network pathways should ensure that an organ forming protein is expressed in particular place and time and space, but not anywhere else. And this PAX6 is present in human, mouse and human too, and relatively conserved. But considering that is so different, you ask the question, really the functionally same. In Drosophila, we know what I told you about PAX6 is required to make the eye. PAX6 should be expressed only in the one place, it should not be expressed in other place. With this kind of an experiment, where people have shown that if you overexpress PAX6 on leg, you get an eye here. You can get eye virtually in any part of the body. I don't know whether Jishi uses this as a lab experiment for undergraduate students, right? So this is what clearly suggests that PAX6 is required, it is sufficient, and also tells you a lot about how the developmental programming happens, right? What Walter Gering's lab did almost 20 years ago, they picked up PAX6 from human and mouse and did the same experiment. So here is the scanning electron microscope of a eye, this is the antenna. In the place of antenna, where the antenna is supposed to be developed, in those cells they made PAX6 to express, and you can see a nice new eye forming here. And you can do this whether you are using a mouse or a human PAX6 gene, suggesting that they are functionally conserved. Now we can ask the same question that a friend asked here. What PAX6 does in human is determining this fate. Of course, it is an eye fate, eye fate, eye fate, but morphologically they are different eyes. Functionally, philosophically maybe it is eye, but it is still very, very, very different eye, right, these two. Then why is forming this kind of eye gene from here and you put it back, right, in the, right, why not some other eye? That depends about finally what actually evolution is all about. Evolution is not really about selecting an organ, selecting very different things. It is only taking really at a micro level. So protein level, its function is a transcription factor, upregulating or downregulating certain set of genes, and those set of genes invariably will also be upregulated, downregulated genes directly or indirectly. If they are transcription factor directly, if there are some signaling molecules indirectly by activating or downregulating transcription factors, and this whole cascade of events, right, finally end up in orchestrating the expression of hundreds of genes, which finally build the eye. If the initial one or two steps are required to specify the place where the eye should be formed, right, and at the same time later events is the one which actually builds the eye. What PAX6 in all these organisms doing is to specify where it is to be formed. So obviously when PAX6 is removed, you see complete loss of eye, whether it is in human or in Drosophila or in mouse. There are very congenital eye defects where the PAX6 is mutant. In human too is associated with eye, blindness. So this is one way of looking at the evolution of morphological features, which now it becomes more complex. We are talking about the physiological features, right? Now if you want to look at the behavior, right, this is even more complex, because behavior is inherently an interaction between the body and the environment, external environment, right. And then I do not want to use the word mind because that is completely misused word, it is unscientific, right. The word, that scientific word for mind is cognition. Now if you want to understand the evolution of cognition, right, and so called consciousness, if such a thing exists, right, obviously if you go on looking at the very complex way various different features have evolved, at some point you expect that cognition and consciousness must have had some roots in, there must be some elements of those evolution of those ones in our behavior or in our network of neurons, in our physiological processes, right? It may be, you know, if you are just comparing the behavior of chimpanzee and human, you should not be able to understand the evolution, right? You may have to look at some other aspect of it, where the signature of that should be able to trace it back to the behavior. So, and there is no doubt that everybody agrees that brain is of course product of evolution, and the brain has a lot of similarity. If you take a chimpanzee brain and a human brain, give it to a neurosurgeon will sometimes get confused, right, whether whose brain it is, kind of stuff. And if you take of course mouse brain, there are a lot of similarities, although it is very small brain compared to a human brain. But there are some major differences also amongst the primates versus non-primate mammalians and kind of stuff. But anyway, so almost everybody agrees that morphological features are product of evolution. Your biochemical and physiological features are also product of evolution, right? Obviously, you know, behavior, at least some of the mechanistic aspect of the behavior will be product of evolution. Behavior per se may not be product of evolution, because behavior, as I told you, is an interaction between body and the environment, and obviously that has to be different between different species and different organisms, different individuals. So, obviously behavior itself, we cannot say in total is a product of evolution, right. The mechanism that of cognition would be the product of evolution. When you say mechanism or cognition, which cognition is the perceiving the outside world and responding, that entire process is cognition. Cognition is not just perception, right. It is not just seeing an image and saying, recognizing this as a face of a human being, face of my mother or a sister or a brother, that is not cognition. That is only a part of the cognition, that is perception. The cognition is the complete process. I am seeing something, hearing something, smelling something, a combination of everything is happening. I am perceiving the world and also responding to the world in a particular way, right. If there is a prey, I go towards the prey. If there is a predator, I go away from the predator. This whole process, perceiving that is a prey or a predator, and then a response to the prey and the predator, is entire process is cognition, right. That has an underlying mechanism to perceive and mount a response, right. What is the response is what we call behavior. It varies from individual to individual and species to species, right. So, if you want to understand the mechanism, I guess the evolution is the only way to look at it, and psychology is not the best way to look into it. So, I will give you a couple of examples of some of the experiments which one or two of them are done in IISER Pune and with the help of some students, and some of them are from the literature. Let us take some basic behavior. Almost all species show is the aggression behavior. Some species show aggression behavior to the members of other species or to the member of other groups. Some of them show members of the same group as the same species, right. For example, honeybees, typically they are aggressive against the members of other species, right, or sometimes to the other members of other hives. I do not know how common it is, right. Same thing with ants also, right. And as long as they are disturbed, you know, but they sometimes they show aggression to their own siblings also, right, depending on their behavior once, right. But in case of solitary insects, right, like for example dogs, they are territorial degree. A group of dogs within themselves they are cooperative. If another dogs come to that territory, they are trying to be very aggressive to that dog. These are solitary insects. Drosophila, for example, is a solitary insect. In nature, they are never live in groups, not even in pairs, the male and female. They are solitary insects. They always happen to be, you know, individuals. So, invariably, is one of their innate behavior is as soon as they see another male, for example, this male or they start fighting with each other, right, either for food or mate, right. Look at this. But of course, this is a happens much faster. Is a fast camera and shown the low speed, and they also go to Rio Olympics sometimes and do the wrestling, and they are wrestling for a female here, and female is happily dead freshly dead, so pheromones are still there, so they are fighting for this female. And so now if you look at one of the cognitive the cognition problems of cognition when you compare different animals and kind of thing is, how do you compare the behavior? Because we always say behavior is a window to our brain. So to see our brain, obviously you have to have some behavior. For example, schizophrenic, you classify someone as schizophrenic based on the behavior. You classify someone as a memory loss person based on the behavior. You classify someone as, you know, sort of, you know, very hungry person based on the way the person behaves and kind of stuff, right? Now this behavior, now if you have to interpret in a particular way, right, and it has to be done in a very objective and scientific way. So how do you sort of define behavior of an insect like this and say because of this is happening? So you need to have a very clear understanding of the behavior. That means you have to have an assay, a behavioral assay, which is reproducible and understandable, right? So one of the major problems of studying animal behavior is, and comparing with human behavior is this one. Now you don't have to even comparing the behavior to behavior. What you are comparing is the mechanism in the brain, what could have evolved in the brain of primates or human, which is not there in other organisms, which could have added an additional complexity of our cognitive abilities, so that we are more complex behaviorally and cognitively compared to other insects, right? So one way of doing this, over the time, you know, the 2-3 labs, including one of our colleagues in ISA Pune, what they did, the experiment is very simple. They, you know, put in this kind of arena, right, one male and female, marked both of them, and they found out which one loses the fight. Typically what happens is, whichever the one loses the fight, it starts running away from the other one, and other one would be chasing it, right, and that is an indicator that this has lost and this has won, right. Now, so there is also a particular time you give. For example, if one is continuously running away from the other one for more than 15 seconds and other one is continuously chasing for more than 50 seconds, you consider this is the, you know, loser and this is the winner. It is like you count in the boxing, right, 1, 2, 3, after up to 10 you count, and if they do not get up, then you say this person is a loser and this person is a winner, right. So that is precisely how it is done. Then you, because you already marked, then let us say this is the loser, you take this and give some rest, some hours, and then put it back in the arena with another male, and then see what happens, and then let us say one of them will lose. Now if you do it this many times with different flies, there is a certain probability that you may pick up a male which has lost consecutively 3 or 4 times, right. Interestingly, this is a phenomena which you know, Martin Heisenberg and others discovered long ago maybe about 5 years ago. They showed that a male which loses consecutively 3 or 4 times invariably starts avoiding the fight itself, right. So suddenly we start thinking an anthropogenic way, right. It's sort of, it knows that it is not a winner, it knows that it gets bullied all the time by others, it wants to avoid the fight, which is typical our behavior too, right. If we are, what, are you the one? You are a lose. Why? You, I know. Yeah, right. So, but you know, without even fighting, you think you are not going to win, right? Without knowing what would be the opponent like. That is precisely what this is also doing. Okay, you are sitting next to him, right? Now, obviously, they also gave the name loser mentality, which is a human name for this, and that loser mentality obviously means, now you can ask hundreds of questions. This is what they had shown, a behavioral paradigm.
How do they control for body deficiency? The fresh male is always going to be in better body condition than the loser because he's fighting many battles. So the subsequent battle is getting.
No. That the experiment was under control, where the males, the body condition means what? And they are generally moving around. Not that there will be a physical damage to the body. You know, typically.
I don't know how much
Yeah.
they fight, but it could be
Could be some, you know, some sensory organs in the antenna may have fallen down or something. 1 or 2 teeth. They don't have teeth, so maybe sensory organs have fallen down. That's what they usually leave them for 24 hours in food and then come bring it back and kind of thing. Yeah. So, well, you can also bring the winners from the other ones. Yeah, that also can do that, right? Or you can also bring 2 males which are both are losers into one. In the one which has, right. So again, this is not programmed, right? This is all, it was sort of has happened. They happen to lose 3 or 4 times, whatever the reason. Then there is a perception of they're losing it. See, this is what is interesting. See, not that you need to have a, so 1 second, not that you need to have your own personal capacity, that what he is saying, because I don't have biceps and this guy has a lot of biceps, and you think that this person is going to win and you perceive that way and then you stop fighting. Let us say it's not that anything, you like kids, what happens, right? Kids invariably don't have very such kind of perceptions, right? A small kid will be thinking that it can win against a big fat person and try to fight. They not only once, they may do that 2 or 3 times, and they realize that they cannot win fight with another person only after losing several times, right? So, having lost some time, you have perceived, you have now a perception of yourself saying that you are a loser, right? And then you start avoiding the fight. That is the behavior they have shown. There are 2 things here. One is, very interesting question is, you are now memorizing an experience, you are not memorizing an image here, right. So, it is not like a conditional memory what people talk about, right. Every time you show a red dot, you get some drop of honey, so that as soon as you see a red dot, something comes out of proboscis or something. Not that kind of a thing. Here you are remembering an experience of losing a fight. So, what is that experience, memory of an experience? It is a very complex cognitive question, which is what haunting for hundreds of years all philosophers and psychologists. Now, you can actually use an insect or a small, and now you can use even mouse and all. This is very interesting. Now, you can want to understand the underlying physiological and morphological mechanism in the brain. You can use an experienced fly to understand this. Okay, and there is no answer yet. It may take another 50 years. I am just posing these questions. The second aspect is, once you assume that it is because it has memorized that it has lost the fight, that is why it is showing the behavior. It has modulated its behavior. Instead of going to fight, it is going away, because it is an innate behavior is to fight, but it is running away to fight is it's modulated its own innate behavior, right. That means it has a memory, depends on the memory. That is easy to test. So, that is they did that. So, what the students did is to take some memory mutants. So, you take acetylcholine receptor mutants which have no memory, long term memory, and irrespective of how many times they lose, they keep on fighting, they go back and again fight, right. It is interesting. So, now it can be used very nicely for these kind of work. That is what I was trying to show. So, I mean, there are many experiments. Flies have been used now, variety of behavioral tests, and advantage is because you can do genetics, you can overexpress genes, you can downregulate genes, you can actually specify in a particular part of the brain. So, you can correlate from molecules to neurons to network to behavior finally, right. And once you have a paradigm, and this paradigm is, let us say, tested in mouse and zebrafish, and if it is similar, and you predict that it could be similar in pyramids too, but there is an added layer of complexity, and that is what we need to understand the human cognition, which definitely is more complex or different. I do not know whether you call it as complex, less complex or more complex. It is definitely different than the other animals. That is why we are all here, right.
Do they have a judgment, like if I put 2 winners, they will fight differently, but if I put 2 losers, do they judge each other, like,
No.
or both of
If
them,
if both of them have consecutively lost several times, they both have loser mentality, both run away.
like they don't have any
No. In fact, yeah, and they also, their decision is not based on the perceived strength of the opponent, like what you are talking about. You have a perceived strength of your left person who is sitting on your left side, but not on the right side, right. So they don't have that kind of perception. For example, you can take a loser and put it with a legless or a wingless or something. Even if it is a half dead male, it will just run away. It will only see as another male and then run away. That's it. Yeah, but we have an added complexity where we can even perceive the strength of the opponent and behave and modulate the behavior, right?
I think this is because of complexity. Even like when a woman is a baby, that time this maturation is not there, like comparing the opponent.
Anyway, there is nothing to do with speciation. Obviously, it is central speciation, but it is too far away removed from the immediate question of speciation. At some point, it will all lead to speciation anyway, because behavioral change, underlying behavioral change, can also lead to reproductive isolation, which automatically leads to speciation. The problem is these flies themselves under laboratory conditions also, they can remain healthy only for about 10 to 12 days. Beyond that, they will start accumulating other problems, age related problems. It is very difficult to do this experiment beyond certain day. And again, what is there, for example, if they can remember for 3 days, maybe it is equivalent to 30 years of us in terms of physiological age, right? That is what I am telling you. They remember for at least, so these kind of experiments are done in such a way that they are remember for few days, not just for few hours.
Is there any experiment done between interspecies, not intraspecies, this kind of fight? But for the chickens and all, those fighting dogs are much more, they have more tendency to fight all the time.
Maybe I will show you one more. Yeah. Now, obviously, I want to understand cognition, you need to understand how information is processed in the brain, because when you say information processed, it is about the sensory input, and also there is a previous sensory input, what you call memory, including our experience, and the combination of that will throw out a behavioral response. So, information processing happens where the sensory input comes to the brain, it is compared with your memory cells and processed, and then a behavior response is mounted. This whole process is your information processing in the context of conventionally what you call thinking, which is not a very good scientific term, right. So, this is the one which is important for our complex decisions, right. Complex decision is, simple decision is, if you are hungry, you eat, right. If you are not hungry, you do not eat it. Second is, if you are hungry, irrespective of what happens outside, you eat it, right. And but at the same time, even simple organisms, not very simple ones, like for example, deer or something you take, if there is a bundle of nice looking green grass and at the same time a tiger next to it, obviously it will not go to the food even if it is hungry, because there is also a predator next to it. The fear of predator dominates over your the hunger, right. And these complex decisions requires obviously complex information processing. You have multiple inputs, right, both positive and negative, and now you have to take a decision. If it is an additive effect, right, there is a nice looking food at the nice smell, both coming, it is an additive effect, right. Now, let us say is two contrasting, one positive, one negative, one repellent, one attraction. So, how do you take decision, right. So, these kind of assays, you need good assays in the animal system if you want to understand which gene, which cell, what kind of neuronal network, and how information is processed, where for example, from visual cord to auditory complex to cortex to olfactory processing cortex, how information gets integrated, how it goes to the memory cells, come back. They're all variety of things one need to do look into it. And again, to the whole point is the method of study, not that answer will come from looking at Drosophila, right? You because in the evolution a pre-existing platform has been modified again and again to give more and more diverse complex organisms. From what we started by saying that a ground state wing programming has been modified multiple times to give it multiple types of insect organ body plan. You can say that a brain once is evolved as a simple brain, few neurons, it has got modified several times, but underlying platform seems to be similar, like neuronal axonal. If you take one neuron, the cell body versus axon and how it is organized biochemically and physiologically, it's very similar from C. elegans to human. And if you look at two neurons and how they talk to each other, the electric signal from the cell body to the tip of the axon, and then post using the synaptic transmission, how two neurons talk to each other, that is also conserved from C. elegans to human. So, some of these underlying mechanisms have not changed. Similarly, at network level too, underlying mechanisms have not changed, but again some modification happened. So, you can now tease out bit by bit from lower organism to higher organism, which is actually not some word that I would like to use. There is nothing like lower and higher organism, from simpler Drosophila to more complex vertebrates, and hopefully you will be able to get some answer in the context of evolution of cognition and consciousness. So, here is a very simple test by a guy called Roland Strauss in Germany. I don't know whether you have met him any time in any conference. Yeah. So, he did a simple experiment. He actually did the experiment for a completely different purpose. He was actually a robotics person. He wanted to see how insects walk, how insects change their directions when they see something, in which direction they have to go, and can you sort of use this whole what you call walking behavior of an insect to design a walking robo, right? And so, basically he wanted to look at the building principles for a robotics using the natural behavior of an insect one. And then he also wanted to see how they avoid or cross a gap or a barrier or a some kind of thing, because if you want to have a robo walking around and doing some work for whatever the purpose where you want to build a robo, you should be able to look at, okay, here is some problem, it has to change its walking behavior and kind of stuff. So, that is the reason he was looking at it, but it has an interesting results later. So, you basically make the insect to walk on a platform, in which this platform is made up of small modular units. You can take out one module, so you can create a gap and see how the insect walks kind of thing. So, he did that experiment, and you see here. You cut the wing, by the way, it cannot fly, so it has to walk. It is the same Drosophila, it is supposed to be flying because it is a fly. So, it senses that there is no solid platform and there are sensory organs on the insect leg. Then it sort of stretches its body, holds on to another side, and then it goes like this. So, a simple way of thinking is, these insects do not know what is solid and what is this one, and how do they know. So, they keep on sort of blindly searching for a solid surface, and it just sits on this, and then it sort of pulls its body towards that one, right, which may be true, and which what sort of thought is true. What it did is sort of increase this. Can you switch off some lights? Oops, I don't know what I switched off. I don't know. Anyway, so he did this experiment with slightly increasing the width of the gap. I mean, you can sort of, I mean, if you make it an anthropocentric view, it's sort of groping and trying to see where this one is, right, fell down because couldn't balance very well. He didn't stop at that point. What he did, he increased the width further, right. This time the fly is coming from that side, a walking fly, not flying fly. See what it does. See, it didn't even make an attempt. I'll come to that, right? Didn't make an attempt. Now, not only that, you take the same original this length, which a normal fly would be able to cross, but you take a blind fly. There are two ways of it. One is genetically blind. Other possibilities, you just put some kind of a a paint which is not too toxic, and then you make it blind artificially, and both of them didn't make an attempt. Even this gap, because as soon as they walking and they saw there was some gap because the front legs didn't sense the solid surface, they just stopped, right? They are walking backwards, right? And then you can take the same fly as your question asked, a fly which is be able to cross here, and when you show this, it does not cross, right. Of course, these are all statistical tests. Nothing like every fly will do exactly the same way. That is an advantage of first of all, behavior is like that, right. Behavior is cannot have a 100% reproducible one, whereas unlike you make a mutation in a gene and you have loss of hair, loss of eye or something, here you need to do more and more statistical ones. The problem with many of our behavioral work in in human is psychologists take 4 subject, 5 subject, 6 subject and then say all population behave like this, right, which is not true, which is a psychology is sometimes they say it is not science, right. But here you can do hundreds of these kind of things. So, you can have a clear statistically significant results, right. Now, what is interesting is, what it means is, it is visually estimating the distance, whether it is crossable or not crossable. Now, if it is to look for a food, whether it has to avoid the gap, you can ask hundreds of testable questions, experimentally testable questions. That is important. You can always ask questions, that our mind can keep asking questions. Unless you are experimentally testable, what is the point? The experimentally testable question is, there is food here, but still it does not want to go to the food because it cannot cross the gap. So, how does it know whether it can cross or it cannot cross, right? And you can ask the question, how does it know what happens if it does not cross? Does it really know something, or it is just some some kind of a hardwired behavior saying that if there is a gap it should not move forward, right? It is nothing to do that it knows there is something danger if it is, right. And we assume that obviously it cannot think that deeply, it is not a philosopher, so it should not know that what happens if it is fallen, right. Now, the next question is, if crossable or not crossable also depends on your own body size. What is, how much distance, how much steps I have to take to touch this one from standing here is different from me, different from another person, different from a child, depending on their body size and the arm length, right. So, that means it should have some understanding of its own body size, which is what self awareness, right. So, that experiment he did. We were discussing this in Pune once, and then he went back and very excited, and what he did, he took insects of different sizes. What you can do, you can grow in under different conditions and make a small and big one. Sometimes you put a crowded wires, you doing like, you are a fly people? Oh, you are doing that, okay. So, you can make smaller ones, bigger ones, and so the what it considers or perceives as crossable, not crossable depends on its body size, right. Then you also notice that the flies which are just born, they tend to make more mistakes, and the flies which are 1 day old or 2 day old, they tend to make less mistakes. That means, the flies which are older, having moved around the world, within a while their own small world, they have an idea of distances, which is also true for us. The human babies, which first few months, they cannot understand the distances. They may be lying on the floor and they think that light should be able to catch, and they try to catch the light, they cannot, and they start crying. Only when they start moving around, crawling and then walking around, they have an understanding of distances. There is an abstract understanding of distance, not like 1 feet, 2 feet, 1 meter and something. So, which is also true for these ones. So, now such complex behavior, which has a obviously a materialistic basis or the basis of the brain, you can now start understanding which part of the brain, what kind of neurons, what kind of interaction, what kind of network, how much, the guiding principles of this kind of behavior also one can study using insects. Okay. I'll stop here.
Any more questions, or we'll write up? Thank you, Shashi. You will be.
Thank you.